if you swallow like 100 milligram pill, you probably just assume your body is about to get 100 milligrams of medicine.
Right. You figure you take the pill and boom.
Exactly. Boom. Your system is now running on this clean, predictable 100 milligram infusion of relief. But what if I told you that depending on the drug, your liver might ruthlessly destroy like 80% of it before it ever even reaches your bloodstream?
Yeah. I mean, biological accounting fraud is basically rampant inside the human body. The amount you swallow is almost never the amount your tissues actually get to use.
Which is wild to think about.
It really is. And the gap between those two numbers, well, that's where some of the most fascinating and, frankly, dangerous medical mysteries happen.
Welcome to the deep dive. Today's mission is to decode this core concept from pharmacology called bioavailability. We've got a great stack of sources today, ranging from foundational medical literature to some pretty wild historical case studies.
Some very wild ones, yeah.
All to answer one deceptively simple question. When you swallow a pill, how much of that drug actually makes it into your system? And let's unpack this. What exactly are we measuring when we talk about bioavailability?
Well, in pharmacology, bioavailability is usually just abbreviated as the letter F.
Okay.
And it stands for the fraction of an administered dose of a drug that reaches your systemic circulation. So, your main bloodstream in its active, unchanged form.
Unchanged.
Yeah, but it's not just about the total volume, It's also about the speed at which it arrives. So to measure this accurately, scientists needed a gold standard to compare everything else against, like a true baseline of 100%.
I would assume that baseline has to be an IV, right? Like an intravenous injection?
That is the convention, yeah. An IV dose is defined as having an F of 1.0 or 100% bioavailability.
Makes sense.
The logic there is pretty straightforward. I mean, an IV needle physically bypasses every single absorption barrier your body has. It just deposits the drug directly into the blood.
So it cheats the system.
Exactly. So every other route of taking a medication, whether you're swallowing an oral tablet or, putting a film under your tongue, wearing a patch on your arm, it's all graded relative to that IV benchmark.
Got it. So if you swallow 100 milligrams of a drug orally, but tests show that only the equivalent of 40 milligrams actually makes it into your main circulation, that drug's oral bioavailability is 40%.
Spot on.
So the obvious question for anyone listening is, where did the other 60 milligrams go?
Right.
Why wouldn't a swallowed pill just simply equal 100%?
Well, because an oral drug has to survive what is essentially a biological gauntlet.
A gauntlet. I like that.
Very unforgiving one. Let's map out this treacherous journey.
Yeah.
When a pill drops into your stomach, it first has to properly dissolve in a highly acidic environment.
Right.
And a portion of the drug just gets destroyed right there by the harsh pH. Then it moves into the intestines, where all these digestive gut enzymes start chewing it up.
So it's taking damage at every step.
Exactly. And assuming the molecule even survives that chemical bath, it physically has to cross the intestinal wall. And even if it successfully makes it across that barrier, it doesn't just flow into your general bloodstream to, heal your headache.
Right. It gets diverted. I actually, I like to think of an oral drug as this VIP trying to get into a super exclusive nightclub.
Oh, that's a good way to look at it.
Right. So first you have the bouncer outside checking IDs, which is the stomach acid and the gut enzymes. Then you have to push through the metal detectors, which represents crossing the intestinal wall.
Yep, the physical barrier.
But even when you get past all of that, you aren't actually inside the club yet. You're funneled into this long VIP hallway, the portal vein. And this hallway leads directly to a final, absolutely ruthless security chief, which is the liver.
A final boss.
Yeah. And this chief just randomly confiscates half your belongings before you're finally allowed onto the dance floor.
That captures the sequential hurdles perfectly. That ruthless security chief process is known in pharmacology as first-pass metabolism.
First-pass metabolism.
Right. Because everything absorbed from your small intestine is forced to travel through that portal vein straight into the liver before it is ever allowed to reach the rest of your body.
And the liver is basically a chemical dismantling factory, right?
Oh, absolutely. It's packed with metabolic enzymes designed specifically to dismantle foreign substances. The most famous group is the cytochrome P450 family.
Okay, P450.
Specifically, this one enzyme called CYP3A4.
Wait, so if a drug is chemically designed to easily slip through the intestinal wall, like, let's say it's highly lipid soluble, doesn't that guarantee it gets into the blood? Like, if the gut absorbs 100% of it, isn't it 100% bioavailable?
What's fascinating here is that this is actually one of the most common misconceptions in medicine.
Oh, really?
Yeah. We naturally assume absorbed means bioavailable, but they're totally divorced from one another because of that security chief we just mentioned. The liver can and will destroy a massive fraction of an absorbed drug before it completes a single lap in your circulatory system. Furthermore, the intestinal wall itself has its own supply of those CYP3A4 enzymes, plus this sneaky little efflux pump called P-glycoprotein.
P-glycoprotein. Wait, that's the one that literally pumps drug molecules back into the gut after they just tried to cross, right?
Exactly.
Yeah.
It functions just like a bouncer physically tossing a rowdy patron back out onto the street. The molecule crosses the cellular threshold, and P-glycoprotein just grabs it and throws it back into the intestinal tract.
That's so frustrating for the drug.
It is. To give you a concrete example from the sources, look at propranolol. It's a really common beta blocker used for heart conditions and anxiety.
Sure.
Propranolol is essentially completely absorbed from the gut. It's often cited at over 90%. It crosses the intestinal wall beautifully, yet its actual oral bioavailability is only about 25%.
Wait, really? So 90% gets through the front door, but only 25% survives the liver to actually do its job.
Yeah, the liver dismantles the vast majority of it on that very first pass. That is the textbook proof that getting absorbed into the body and surviving to reach the general circulation are two vastly different hurdles.
So if the liver and the gut are the main obstacles here, does the physical design of the pill actually matter? Or is bioavailability just a fixed chemical property of the drug itself? Like, is propranolol just always 25% period?
Well, for a long time, the medical field actually viewed it as a constant. Early pharmacology textbooks wrote about bioavailability as if it were this fixed molecular property.
Like the boiling point of water or something.
Exactly. But modern biopharmaceutics has completely rejected that framing.
It's a massive conceptual shift because today we know bioavailability is not just a property of the drug. It is a property of the drug, plus the specific formulation of the pill, plus the individual patient's biology.
Right. And to measure that complex, three-part system, pharmacokineticists insist that bioavailability isn't just one number, it's actually two numbers glued together.
Okay, two numbers.
You have extent and rate. Extent is how much of the dose eventually gets in. We measure this mathematically as AUC, or the area under the curve on a blood concentration graph.
Right.
Rate is how fast it gets there. That's measured by Cmax, which is the peak concentration, and Tmax, the time it takes to hit that peak.
I think visualizing that is a bit tricky for anyone who doesn't, look at graphs all day. Let's try this. Think of Cmax as how hard a rainstorm is downpouring at its absolute peak.
Okay.
That's the intensity. While AUC, the area under the curve, is the total amount of water that actually accumulates in the rain barrel over the entire day.
That's a really great way to visualize it. And both of those metrics are critical for you as a patient. Total exposure just isn't enough. The timing has to be right.
Because if it's too fast.
Right. If you take a strong sedative and the rate is too fast, so that massive downpour, you get this sudden immense spike in your blood, a huge Cmax. That can actually cause dangerous respiratory depression.
Right, which is lethal.
Exactly. But on the flip side, if you take a rescue inhaler for an asthma attack and the rate is too slow, waiting 40 minutes for the drug to kick in makes it clinically useless, even if the rain barrel eventually fills up.
Here's where it gets really interesting. The realization that manufacturing deeply impacts both of these numbers, it didn't just happen in a sterile lab. It took a major public health scare. In our sources, we have this incredible event from 1971 known as the digoxin crisis.
Yes. So digoxin is a medication used for heart failure and irregular heartbeats. It has what we call a narrow therapeutic index.
Meaning the margin of error is tiny.
Very tiny. The window between a dose that helps you and a dose that poisons you is very, very small.
So back in the late 60s and early 70s, doctors were prescribing digoxin and they started noticing something terrifying. Patients who had been perfectly stable on the drug were suddenly coming into the hospital with either severe toxicity, meaning their hearts were being poisoned, or therapeutic failure, meaning the drug just stopped working entirely. But when the doctors checked the prescriptions, the patients were taking the exact same milligram dose they always had.
The exact same active ingredient, same labeled dose on the bottle, but they were from different manufacturers. So in 1971, a physician named Sidney Lindenbaum and his colleagues published this landmark study in the New England Journal of Medicine. They tested different commercial brands of digoxin tablets on the same patients.
And what did they find?
The data showed that some brands delivered roughly twice the systemic exposure of others.
Twice the exposure from a pill that said the exact same milligrams on the bottle And the culprit wasn't some grand conspiracy either. It was almost absurdly mundane.
Incredibly mundane, yeah.
The researchers found that differences in the tablet's particle size and the pressure the manufacturer used to compress the powder into a pill completely changed how the digoxin crystals dissolved in the patient's gut.
Think about the difference between trying to dissolve coarse sugar cubes in a glass of iced tea versus fine powdered sugar.
Oh, the powdered sugar dissolves almost instantly.
Right, because it has massively more surface area exposed to the liquid. By tweaking the particle size of the digoxin, some manufacturers were basically creating powdered sugar and some were creating sugar cubes.
Wow.
That completely changed how much of the drug actually survived the biological gauntlet. Nothing about the drug molecule itself changed, only the manufacturing.
This crisis was a massive wake-up call. It led to the 1977 FDA regulations, driven by the founders of pharmacokinetics like Gerhard Levy, John G. Wagner, and Milo Gabaldi.
Yep.
They basically forced the government to step in and say, look, you can't just prove a generic pill has the same chemicals in it. You have to prove it is physiologically equivalent in the human body.
They established the mathematical machinery, like those AUC and Cmax comparisons we talked about, that turned bioavailability from this vague clinical guess into a hard number you could actually regulate. They proved definitively that formulation determines outcome.
Okay, so if the physical pill matters that much, what happens when the patient inadvertently introduces a new variable into the system?
Oh, this is a great pivot.
Let's talk about grapefruit juice, because this is one of the ultimate biological hackers.
It really is. This brings us to one of the most famous accidental discoveries in pharmacology.
So in 1989, a Canadian researcher named David Bailey was running a study. He wanted to see if drinking alcohol interacted with a calcium channel blocker called felidipine, which is a blood pressure medication.
Right.
To run a proper double-blind study, he needed a way to hide the taste of the alcohol from the volunteers. So he just used grapefruit juice as a flavor masking trick. It was literally just meant to be a placebo disguise.
But the results were staggering. In the group that drank the grapefruit juice, the blood levels of felidipine shot up dramatically. And it had absolutely nothing to do with the alcohol.
Bailey had accidentally hacked the first-pass metabolism system.
He really did. It turns out that grapefruit juice contains these natural compounds called furanocoumarins.
Furanocoumarin?
Yeah. And these compounds essentially act like a biological superglue. They bind irreversibly to those CYP3A4 enzymes in your gut wall, the ones responsible for breaking down the drug.
Oh.
They just glue the enzymes shut, rendering them completely useless until your body can build brand new ones, which takes hours or even days.
So the grapefruit juice basically knocked out the bouncer at the club.
Exactly. By disabling that part of the first-pass gauntlet, massive amounts of the drug that normally would have been destroyed were suddenly allowed to flood straight into the bloodstream.
For you listening, this isn't just some quirky piece of trivia. This accidental flavor masking shortcut is the exact reason why, when you pick up a prescription at the pharmacy today, you might see a bright yellow sticker that says, "Do not take with grapefruit juice."
Yep, you see it all the time.
It applies to dozens of medications, including several statins for cholesterol and various blood pressure meds. Because if you drink grapefruit juice with them, you aren't getting the prescribed dose. You're getting a massive, potentially dangerous overdose.
It perfectly illustrates that bioavailability is a living, dynamic property.
I mean, it changes based on what you ate for breakfast.
So, knowing how fragile and easily manipulated this system is, we have to look at how governments handle generic drugs. Going back to your earlier point, how do regulators ensure that a cheaper generic actually works the same as the expensive brand name pill?
Right, because after the digoxin crisis, the FDA set up rules for bioequivalent.
Yes. The regulatory math works like this. The FDA and the European Medicines Agency state that two products are bioequivalent if the 90% confidence interval of their AUC and Cmax ratios falls between 80% and 125% of each other.
Hold on a second. Let me translate that math for a moment just to make sure I'm hearing this right. You're saying the government considers a generic drug equivalent even if it gives you up to 20% less or 25% more exposure to the drug than the brand name.
The numbers bear that out, yes.
Yes.
It's a major misconception we really need to clear up. This is misconception 3 in our sources. Bioequivalent means the exposure falls within a statistically defined window, that 80 to 125 range. It does not mean they are literal chemical clones in how they behave in your blood.
That sounds incredibly concerning.
Well, for the vast majority of drugs, say, ibuprofen or a standard antibiotic, That window is perfectly fine. Your body won't even notice a 10% swing in absorption for a headache pill.
Sure.
But this statistical window is a massive ongoing flashpoint for those narrow therapeutic index, or NTI drugs we mentioned earlier,
Yeah.
where a tiny shift in blood levels can mean the difference between health and disaster.
Yeah, the sources list drugs like warfarin for blood clots, levothyroxine for thyroid issues, and phenytoin for seizures.
Right. Neurologists have been sounding the alarm on this for decades. There are published series of patients who've been perfectly stable on a brand-name anti-epileptic drug for years.
Hmm.
Then their pharmacy switches them to a generic, or even switches them from one generic to a different generic, and the patient suffers a breakthrough seizure.
Oh my God.
And the frustrating part is, both of those generics technically pass the FDA's bioequivalence testing. They just sat on opposite ends of that 80 to 125 spectrum.
It's just enough of a difference to cross the threshold in that specific patient's brain. And there's also the famous case of cyclosporine, right? The anti-rejection drug for organ transplants.
Well, the Sandimmune to neural reformulation, that is a classic example. So the original drug, Sandimmune, had highly erratic bioavailability. It depended heavily on what food the patient ate and, their bile production.
Okay.
So the manufacturer created a new microemulsion version called Neoral that absorbed much more predictably. But you couldn't just swap them out milligram for milligram. Carelessly switching a transplant patient between these formulations pushed some patients into acute organ rejection or kidney toxicity.
That's terrifying.
It is. Same drug is not remotely the same thing as same bioavailability. This raises an important question about how we test these drugs today, actually. There's a huge debate around something called the Biopharmaceutics Classification System, or BCS.
Now, what is that exactly?
It's a framework that sorts drugs based on how well they dissolve in water and how well they cross the intestinal wall. The debate really centers on something called biowaivers.
Biowaivers, meaning they literally waive the requirement to test the drug in humans.
Exactly. For certain highly soluble, highly permeable drugs, regulators will sometimes allow manufacturers to skip expensive human trials entirely. They just do lab tests to see how the pill dissolves in a beaker of simulated gastric fluid.
Just a beaker.
Right. And advocates say this saves millions of dollars and gets generic drugs to market faster.
But skeptics argue that a glass beaker dissolving a pill in a lab doesn't perfectly mimic human gut motility. I mean, a beaker doesn't have a liver, or fluctuating enzymes, or a stressed-out nervous system that changes how fast your stomach empties.
We see this all the time. Laboratory dissolution is just a proxy. And sometimes the proxy completely fails to capture the complexity of human biology.
So what does this all mean for you, the listener, navigating the healthcare system? How do these pharmacokinetic principles shape the decisions your doctor makes every single day?
It shapes everything, often invisibly. For example, if someone is having a heart attack and needs nitroglycerin for chest pain, why do we give it as a spray or a tablet under the tongue? Why not just swallow it?
Because of the gauntlet.
Exactly.
Yeah.
Because nitroglycerin's oral bioavailability is incredibly low due to first-pass metabolism. Putting it under the tongue, the sublingual route, allows the drug to absorb through the mucous membranes directly into the systemic circulation, completely dodging the liver on the way in.
It's the secret back door to the club.
Yeah.
You bypass the bouncer and the security chief entirely.
Yes.
And this also explains why patients with liver cirrhosis need radically different doses. If your liver is impaired and heavily scarred by disease, that ruthless security chief is suddenly asleep on the job.
Right. The bouncer is gone.
An impaired liver lets unpredictably high amounts of an oral drug slip through into the blood, so doctors have to drastically lower the dose to prevent accidental overdoses.
And this leads us to two more critical misconceptions. Misconception 4 is that higher bioavailability is always better. People just naturally assume you always want maximum absorption.
But you don't.
Far from it. Extended-release opioids and certain blood pressure medications are deliberately engineered to have lower, slower bioavailability. The manufacturers intentionally blunt the absorption to avoid a massive spike, a dangerous Cmax.
Right.
The goal isn't maximum exposure. It's creating the right pharmacokinetic profile over an extended period of time.
That makes total sense. And misconception 5 is the idea that because an IV gives you 100% bioavailability, it somehow guarantees the drug reaches its target.
Right. 100% IV bioavailability only means it reaches the general blood supply. It says absolutely nothing about whether the drug can actually cross the blood-brain barrier to reach a tumor or penetrate deep into bone tissue to fight an infection. Systemic availability and target tissue availability are two very different hurdles.
Which brings us to perhaps the heaviest, most widespread real-world consequence of all this, the opioid crisis. Bioavailability is at the absolute center of how we manage pain in this country.
It is. Take oral morphine, for instance.
Mhm.
Because the liver rapidly processes it, its oral bioavailability is only about 20 to 40%.
That low.
Yeah. The mechanism here is called glucuronidation. As the morphine passes through the liver, enzymes literally attach sugar molecules to the drug. This tags the morphine, making it highly water-soluble so your kidneys can easily filter it out into your urine.
Wow.
Because the liver tags it so fast, to get the same pain relief as a 10 milligram IV injection in the hospital, a patient might need to swallow 30 or 40 milligrams of pills at home.
The problem is that governments and pharmacies track opioid prescriptions using a metric called morphine milligram equivalence, or MME. It's a national calculation used to legally limit how many opioids a doctor can prescribe you, basically tracking your total exposure.
But those MME calculations rely entirely on fixed mathematical assumptions about relative bioavailability. Pain specialists have heavily criticized these charts for being completely oversimplified.
Right.
As we've discussed today, bioavailability isn't a fixed math equation. It changes based on genetics, gut health, and formulation. Because the MME system treats bioavailability as a hard, rigid rule, these pharmacological debates are directly impacting who is allowed to get pain relief and how much they are permitted to receive.
It's incredible. We started this deep dive talking about the assumed simple math of taking a pill, but the reality is that swallowing a medication is this complex, high-stakes negotiation with your body's biological gauntlet.
Constant negotiation, yeah.
Bioavailability isn't just a number in a textbook. It's a dynamic, living property made up of the drug molecule, the manufacturer's tablet press, and your own personal biology.
If we connect this to the bigger picture, from the digoxin crisis that changed FDA regulations, to the accidental discovery of grapefruit juice acting like superglue on our liver enzymes, the fraction of the dose that actually reaches your circulation is a delicate, constantly shifting balancing act.
Which leaves us with one final provocative thought for you to ponder. We've seen how deeply bioavailability is dependent on the patient's system, and we know that our gut motility, our liver enzyme activity, and our diets fluctuate daily based on our stress levels, our sleep, and the meals we eat.
Yeah.
Does that mean the actual usable amount of medicine you absorb from your daily prescription is slightly different every single morning?
Well, it's a fascinating and honestly a slightly unsettling reality of human biology.
It really is. Thank you for joining us on this deep dive into bioavailability. Keep questioning the science behind your everyday lives, and we'll see you next time.